We should move on to the next speaker, who's Victor Greiff from University of Oslo, where he's an associate professor of immunology. And without further ado, Victor, please tell us.
Yes, thanks so much for the invitation. I hope you can hear and see my screen clearly. So I'm going to talk about immune receptor analysis for diagnostics and therapeutics discovery. And I'm going to show you a basic overview of what immune receptor analysis coupled with machine learning could do in the current crisis that we're facing. So just to catch us all up, what the adaptive immune system is and what adaptive immune receptors are. So any one of us who encounters immune events over our lifetime, for example, infections, disease, and vaccination, all of these immune events, which how I'm going to call them now, are encoded actually into our adaptive immune system as immune memory. And this immune history is specifically encoded in the immune receptors that are membrane bound to the T cells and B cells. And those receptors on the B cells are also called antibodies. And B cells and T cells together are called the adaptive immune system. So if the adaptive immune system is a flash drive that records all past and current immune events, then of course this is very much important for finding new vaccines and diagnostics. So why is the analysis of the immune repertoire so complex? So if we looked at the entire human population and beyond, the potential immune receptor sequence diversity is larger than 10 to the 13, and the exact number is unknown. The sequence diversity in each human of B and T cell receptors is about 10 to the 7 to 10 to the 9. And the sequence length of each of those receptors is about 110 amino acids, but only a small portion, which is called complementarity determining region 3, about 15 amino acids, is actually the most important for binding the antigen. And there have been two main applications in the field of immune receptor analysis, which is using this immune memory of immune receptors on the repertoire level for diagnostics. So for using computational approaches and machine learning approaches for differentiating between or predicting health status, immune status. And on the sequence level, there is also a huge effort both by industry and academia to define novel computational approaches for in silico designing antibodies against a given target, which, for example, here it's called antigen, but it could be, which is a general name for vaccine, bacteria, viruses. And just to give you an impression of the scale of this market, so by 2025, the monoclonal antibody market, which is mostly used now for cancer therapy and autoimmune disease therapy, is estimated to be about $200 billion. So it's a highly interesting market, both science-wise, but also economic-wise. So to make it a bit more specific, what I mean with immune receptor sequencing and how the whole workflow is. So an adaptive immune cell, here a B cell with an antibody, but it could be a T cell as well, binds an antigen, and the receptor sequence is encoded in the DNA, which makes it then very simple to sequence from the blood or other organs via a normal high throughput sequencing. And then per individual, you get high throughput sequencing data sets, which are here by these little lines. And the problem in our field so far is that most of these data are unlabeled. And by unlabeled, I mean that we know the label of the data set itself. So we know if it comes from a healthy or a diseased person, but we don't know to what antigen these receptor sequences actually are specific to, since we lose the link when we do sequencing between the receptor and the antigen. So we actually then need machine learning approaches to try to find those sequences that are disease associated. And the biological problem is that these immune receptor sets in our body are huge. The data sets that we get from the sequencing are huge. And but the incidence of disease-associated sequences is quite small. So it can be as low as 1 in 1 million cells and even lower than that. But this depends on the disease. So the machine learning, the ideal machine learning workflow of what the immune receptor community is dreaming right now is that if we had patients of which we sequence the repertoire, where we don't know what the sequences are, what they bind to, and so that we then perform classification in order to then understand which individual has been affected by which disease in the past and also currently. This is, of course, very important for checking vaccination status, for checking autoimmune disease status, cancer status, etc., etc., etc. And via this feature selection, then, which can, for example, be done by multiple instance learning, which we have just co-published with the Günther Klammbauer lab. It's just on bioRxiv. It is possible then to then find a posteriori back those sequences that are associated with disease, and then you could follow up further on those. Could they be used for therapeutics, or in autoimmune disease, do we have to knock them out because they are disease driving? So here, I think it's a huge untapped market for machine learning in general. And I think the immune receptor community would be very grateful if more machine learning groups would hop onto this very, very challenging problem. On the sequence level, the problem is equally challenging. So we have here, for example, the use case would be we have an antigen and an immune receptor. Here it's an antibody, and they interact via molecular interactions. And then the problem is, given an immune receptor, here an antibody and an antigen, for example, a vaccine, would the antibody bind the antigen? And if they would bind, where on the antigen surface would that bind? And this is called epitope prediction. For antibody, this is mostly unsolved. For T cells, it's a bit solved, but still very much overall not solved. Then for vaccine design in general, the huge problem is, can we make a vaccine such that our pre-existing naive immune repertoire would be able then to actually bind the vaccine if it's injected into our body. So, and this would open the door to personalized vaccination therapies, depending on the pre-existing antibodies that we have in our system, or T cells that we have in our system. So these are huge challenges where machine learning is, I think, greatly needed. And just to catch up or to summarize what the main challenges are that we are facing that are also interesting for the machine learning community is that immune receptors, I told you, per individual, so the repertoires, we have 10 to the 7 to 10 to the 9 per individual, and they're mostly non-overlapping across individuals. So every one of us has a unique fingerprint of immune repertoires. Each repertoire can, of course, have potentially many labels, because we can have many different diseases, yes or no. So it would be very nice to predict all of these. And then there are also, as Fabian also has mentioned, there are confounding factors here as well, such as age, sex, and genetic background. And then, of course, we would not only be like to classify, we would also like to recover those sequence patterns that are actually driving disease to then follow up with this therapeutic approach. On the sequence level, I think the main complicating factor is that since we only have 10 to the 7 to 10 to the 9 antibodies in our system at any one time, and the antigen space is much, much larger, we need to be able to, one antibody needs to be able to bind many different targets. So one antibody cannot only bind one antigen, but many different antigens. And each antigen can also be bound by a multitude of immune receptors. So it's like a high-dimensional two-body problem, making machine learning also much more difficult. But the main problem that we have so far is this lack of labeled data on the immune status level. So we lack we lack large-scale data sets on the repertoire level and large-scale data sets on the sequence level where we know what antigen a given immunoreceptor sequence is binding. And this is an experimental issue and a money issue. So what we actually need is a Rosetta Stone for immunology, where to each immune cell we could unambiguously attribute the antigen. Here, this was pre-pandemic. This was for a blog post. And here we would like to, for example, link immunoreceptor sequence to a given flu strain. If this could be possible just by sequencing and then machine learning, that would be a huge advance. So, but all is not lost. There are novel technologies surfacing right now. And these technologies are spearheaded, for example, by 10x Genomics. And they have just published last year a technology where you can, at high throughput on the repertoire level, label with the antigen T cells and also B cells. And also academic labs are working on droplet approaches for linking immunoreceptor sequence to function. And there is also a huge effort by Adaptive and Microsoft. So Adaptive is a very large immunoreceptor sequencing company, and they are expanding their TCR antigen map to the COVID-19 immune response, so that they can actually map antigen data to T cell data. And there are also ongoing high throughput in vitro approaches where people like to actually understand what are the long range dependencies that actually enable binding or not binding. So this might be of interest. So there is this AIR community, so Adaptive Immunoreceptor Repertoire community, and I'm actually on the executive board on this community. And there are many, and the aim of this community is to standardize adaptive immunoreceptor research, which is of course very important for machine learning, because you want to have standardized data read, and there are many working groups. They're working on standardizing different parts of the immunoreceptor software pipeline. And we have published these standards also in many papers. Here's one paper in Nature Immunology. And we have also recently issued a call that people that have immunoreceptor data, that they put them online as soon as possible, so that everybody can have access to them. And for now, there are 2 or 3 data sets only available that have immunoreceptor data, but I think in the next month, we will see a very large increase in those kind of data sets. And then we can apply all of these computational and machine learning approaches that I just told you about. So there have already been efforts for machine learning approaches on immunoreceptor data. So we and others have previously published ground truth data simulation frameworks to kind of make up for the lack of labeled data, so that we can at least benchmark machine learning approaches on data sets that have the scale and the complexity of experimental immunoreceptor data. It has been shown in the past, for example, by adaptive biotechnology, natural genetics, that CMV status can be almost perfectly predicted based on a T-cell receptor repertoire. And we recently replicated this also together with Klambauer Lab, who had spearheaded this approach using attention-based multiple instance learning. There had been some progress on prediction of antigen binding using machine learning, and also a quite novel thing is to generate new antibody sequences using autoencoders and other approaches for making new antibodies. So how I think ELLIS and the AIR community could collaborate is via this diagnostics working group that we have, where we are specifically looking for statistical and machine learning approaches for pattern recognition in immunoreceptor data. And as I said, there are a few data sets available, but I hope in the future there will be many more. And many countries are establishing large-scale biobanks, which will enable large-scale immunoreceptor sequencing. So just to 1 more minute, just to quickly summarize recent progress in our lab. So we are currently working on a large-scale machine learning platform that will enable standardized machine learning and reproducible machine learning on immunoreceptor data, which I think will also be very important for the machine learning community that would like to hop on this immune receptor bandwagon, because you don't have to know as much about the biology if everything is standardized with read-in and pre-processing, etc. So this will be hopefully available this year. And then we also recently put out a bioRxiv preprint where we actually showed for the first time using network theory and machine learning that antibody-antigen binding is predictable and hopefully will be better predictable once more data comes along. And we also summarized all of these outstanding challenges for immune receptor analysis and machine learning in a recent review. Yes, and with that, I would like to thank my lab and our collaborators, and especially Gecheta Sandra, with whom we are collaborating very, very closely on all of the research topics that we are having with immune receptor analysis, and also the BGI, Todd Rusko, Engeleda Miho, and also Günter Klambauer. And thank you very much for your attention.